All right. Welcome everybody. It's a great pleasure to see you all and be here for the first GNPS user meeting. It's pretty cool. Before Ming and Peter are gonna kick off the meeting, just a couple of technical details. So I just posted the link for the Google document where we have all the meeting information in the chat. So please feel free to open it. You will see the program as well as abstracts in there. And there's also 3 additional Zoom links for our breakout sessions later. So as we want to have this meeting as interactive as possible, we will have breakout rooms for all 3 sessions where after the meeting, you can just like talk and a little bit more like in detail with like the speakers and ask more questions. During the meeting, we would ask you to ask questions mainly through the chat, and then the session chairs will read these questions for you, and then eventually unmute you if there's like more follow-up and we want to have like a little bit more of a discussion. So yeah, welcome everybody. Thanks for stopping by, and I give over to Ming for the welcome.
Thank you, Danny. Thank you. And so I don't have much to say, but I just want to thank you all for coming, and thank you for the organizing committee. Danny, Julia, they've been the main players. I don't actually do anything these days. But again, the community has been growing and has been very supportive. And when we started this kind of endeavor 7 years ago, something like that, we never imagined that it would grow to such a big community. But we are so happy that it has, and it's because of that. We think the tools have become so powerful. And it's also very motivating for at least us, the core team at San Diego, that we actually want to develop tools. It's for you all. And so without further ado, I'll have Peter give the opening talks or opening remarks, just to give a little bit of background on GNPS and where we started from. But it should be quick, and we'll get right into the speakers. Well, anyway, thank you so much for coming, and hope you enjoy.
Alrighty, thanks for the introduction, both Daniel and Ming. For me, what I really like about GNPS is that it's really the goal that we're democratizing mass spec analysis. And when I look at the registration, and those of you who are on Zoom or YouTube that joined today, I just get really excited. And I think Ming nicely illustrated it, is we never imagined that we would have so many users. But not only do I see developers that are on Zoom today or on YouTube today, but also users, future users, and even some of that are just getting to know us. But what I really see in many ways is the future of mass spec analysis, but also the future leaders that take this science to the next level. We provide an infrastructure, but it's you guys that really are the future of where these tools are going to go. I think most of you by now know that, but not everybody. I think GNPS is not really an ordinary platform. It was specifically built with community in mind, and that's a bit of an unusual way of how people do science or develop tools. It has had, in many ways, really heavy input by users that are testing and continue to be shaped by the needs of this particular community. For me, this democratization is super, super important, because it means that no matter who you are, where you live, what type of compute capabilities you have an opportunity to do mass spec analysis at the highest level of science possible. So a high school student versus a high level professor at a major research institute backed up with millions of dollars of research grants, they can do the science at the same level. And so to me, that you don't have to worry about compute costs, that you don't have to purchase costly reference libraries, that you don't have to purchase costly software is really transforming, I think, how people communicate and interact with each other. And I think one way to think about GNPS is really that it's built for and by the community. And I say that because we've now more than 50 people worldwide that have created code for GNPS. It has more than 100,000 users. I actually don't know the current number, but it has a large user base. They've contributed 400 million MS spectra into the public domain, and I think this will double every 2 years based on the current trends. The community has provided 150,000 reference spectra. This is your knowledge. This is the knowledge that each one of you who uses GNPS, that's what you use. And so I hope everybody else contributes to it, because then you're contributing to the democratization of science. It's now accessed 200,000 times a month by 160 countries. And it's even taught now in high school and undergraduate classes. To me, this really is sort of the epitome or exciting aspect of being able to democratize science, that people all the way from high school to very advanced science labs that are able to really do high-level mass spec analysis. And so in many ways, the future is already here. And I could not envision the kind of science that the users are going to be talking about today just 4 years ago. And I imagine that some of the people who are going to be sitting in on Zoom or YouTube, they're also going to be looking at some of the talks and going to say, oh, I can use it in this way. And again, these are not things that we envisioned. So thanks again for being a really great community to contribute to the democratization of science. But I'm handing it off to Fidel and Audrey at this point, because they will introduce the next speakers.
Thank you, Peter. We start the first session, which is metabolomics, and my name is Fidel Tugizimana, based in South Africa, University of Johannesburg, and Omnia. I do mostly plant metabolomics, and I will pass to Audrey to introduce our first keynote speaker for the session. Audrey, the floor is yours.
Yeah, hi everybody. So good morning everyone, good afternoon everyone. Could I have your attention please? I'd like to welcome all here today. My name is Audrey Le Gouelec, and I'm an associate professor and hospital practitioner in Grenoble in France. And today our first keynote speaker will be Mehdi Benidir, an associate professor from Université Paris-Saclay in France. And he will have 12 minutes for his talk. And before, I want to briefly introduce him. So Mehdi pioneered the use of molecular networks for natural plants, in particular for the discovery of structurally complex alkaloids, and also contribute to the development of anticipation-based annotation methods. And in today's meeting, his talk is titled Harnessing Chemoinformatic Tool for the Discovery of Natural Products. So welcome Mehdi, and now you are on.
Okay, thank you, Fidel. Thank you, Audrey, for this kind introduction. Thank you so much for the organizing committee to select me for opening this first GNPS user meeting. I'm very, very excited to do that. During this talk, I would like to share with you our recent research results dealing with the discovery of natural products using chemoinformatic tools. As you can see from this slide, I come from a natural product chemistry team with a strong interest with different sources of natural sources. We have plants historically, and recently we have expanded our attention to marine invertebrates, microorganisms, and micromycetes. Over the recent years, the development of the analytical tools and the bioinformatic tools led us to develop what we can call an MS-based integrated diversity mining pipeline. The latter starts with a natural source, its extraction, a profiling using LC-MS. We obtain this so-called raw data that will be processed using the so-called feature-based molecular networking. And I would like to take this opportunity to congratulate all the authors that are behind this beautiful contribution to the community. Using Gauss in mind, we harness the MS1 data to create bit lists. After that, we obtain molecular formulas, and we search these molecular formulas against classic natural product databases such as DNP, Reaxys, or MarinLit. At this stage, we have a first level of dereplication. We also take advantage of the MS2 data that will be organized using the GNPS platform to build molecular networks. Molecular networks will be annotated, okay? We have in-house databases. We also use all the annotation tools that are presented in the website, such as Network, MS2LDA, or ISDB, to name a few. And from all this data, we try to build a complete scenario of a finely tuned scenario to isolate new natural chemical entities in an efficient and streamlined way. Of course, we have also an interesting and main expertise in our group dealing with structural elucidation and absolute configuration assignment. Okay, folks, we are now in front of what we call a molecular network, okay? So from a blind standpoint, this network can be considered as a roadmap. This roadmap needs to be further enriched with spectrometric annotations and functional annotations. And deep in the DNA of natural product chemists is the desire to harness the signal of structural novelty among these nodes, okay? And this is what we are going to do in our group. And we have summarized a lot of examples in our recent review in natural product reports. Interestingly, the quest for tackling the issue of molecular networking annotation has fueled a number of successful and creative endeavors in our group, starting by the discovery of monoterpene indole alkaloids using an in-house database that we have shared with the community. And we also used MS2LDA really recently to discover new compounds. We also applied these tools to the discovery of a new blue chromophore from marine natural products. We also used the replicator to expand the peptidic composition of cyanobacteria. With the help of Grégory Jantajouve, we also take advantage of network tool to expand the annotation to what we call the in silico metabolization. And we conceive the CAMPA approach called computer-assisted natural product anticipation. As you can see, this structure here, this anoxide alkaloid, its structure has been totally anticipated before its isolation using this tool. And very recently, we were able to bridge molecular networking and organic chemistry to unveil the artifactual origin of these black tea alkaloids called camelimidazoles. Of course, I don't have the time to tell you all these stories, and I will focus my attention on my favorite area of research dealing with the monoterpene indole alkaloids. Okay, folks, fasten your seatbelts. We are going to the monoterpene indole alkaloids chemical space. Honestly, for me, it is a breathtaking cabinet of curiosity. If you have a look for all these very nice structure that have totally enhanced the pharmaceutical industry. In terms of biosynthesis, monoterpene indole alkaloids derived from this unique precursor called streptozidine, okay? And this family includes more than 3,000 compounds described. I can show you here something like 42 main skeletons that can be encountered in this family of alkaloids. And beside these monomers, this family can be further enlarged to far more complex dimers. As you can see here, you have here the example of venblastine, which is a famous anti-cancer drug. For now, the most complex structure in this family of alkaloids can be traced back to alasmontamine A, which you can see here in the center of this slide. You can see here the CC bond that bridge the two dimer of this family of alkaloids. So in our lab, with all this love for these natural products, we have decided to build a database based on the history of a lot of laboratories from France, Belgium, Spain, and Malaysia. And we decided to share this database with all the community. And I would like to thank Peter and Ming for facilitating the uploads on GNPS. We also shared the database on metabolites, and we have now something like 172 compounds covering 30 skeletons of this family of alkaloids. You can have a look on the GNPS public spectral library. We have our contribution here. And recently, we also have another contribution to this library by adding the lichen database. Let's go back now to jungle, and I will show you a lot of plants here that we have recently studied in our laboratory. And we have decided to launch what we have called the Revisiting Previously Investigated Plants. This is my Hall of Fame of molecules that we have discovered recently in our lab. And for this talk, I will just focus on the recent example of Callichilia inaequalis. Callichilia inaequalis is an Apocynaceae plant native to West Africa and known to possess antiplasmodial properties. We reasoned for this project to use MS2LDA and the power of the annotation of molecular networks using the GNPS spectral library. You have here the network alkaloid extract of Callichilia inaequalis. The red notes here are what has been dereplicated against the GNPS spectral libraries. And as natural product chemists, we try to detect and to select the cluster that are non-annotated and that finally possess peculiar substructure signature. For this work, we have used all these tools, GNPS, AmSinMine, MS2LDA, and our database. And if you have a look to the network for the cluster A here, using MS2LDA, and I would like to thank here Justin for his help, we were able to detect MS-MS spectra with specific mass to motif related to 9 nodes of this molecular family. Here we have an unknown mass loss, and as natural products, we like unknown things. And unknown mass loss that has not been dereplicated against any MS2LDA experiments, another motif DB. So at this point, we have decided to launch the MS-guided isolation and structural elucidation toward one of these nodes. And we finally obtained these 2 compounds that we have called inaequalisins. As you can see, these compounds are new and possess a very interesting structural feature. Let me tell you why. If we have a look to the biosynthetic pathway of this compound, you have to know that there are really limited biochemical pathways that allows the dearomatization of indoles into indolines in the C3 position. Known pathways encompasses methylation, prenylation, and oxygenation. And here with this example, we are proud to present another example of dearomatization unit using the phenylpropan scaffold. This is one fundamental aspect deriving from the natural product discovery process, is finally the generation of puzzling biosynthetic question. Okay, I come now to give you some messages to finish my talk. I'm sure that our generation of natural product chemists is totally gifted to live this exciting era of natural product targeting following finely tuned scenarios. And I would like to emphasize the importance of the community effort, because by the help of all the documentation and that are connected on the GNPS, all these tools are going mainstream, and this is very important for the community. Another point to finish my talk is to never forget that natural products have a long-lasting history in challenging state-of-the-art analytical techniques, and we have to use these compounds to challenge all the techniques. Okay, the most important slide of my talk is to thank all the folks that make all this work possible. Hardworking students, and I'm totally indebted to them. Thank you for your attention.
Thank you, Mehdi, for this very interesting talk. There is no question on the chat, so I don't think. I don't see the question. Maybe some of you have seen the question. I think I have one question. Did you try the feature-based molecular network, or did you want to try these tools for your research? Because I think it will be very interesting to test this new tool of GNPS for your research.
Absolutely, we are using FBMN for our work because we have a lot of isomers in our work, and for natural products, it's very important to use this feature. We are using it.
Okay, thank you. Maybe Fidel, you could introduce we can thanks again Mehdi for this fabulous talk, and now Fidel, you can introduce the next short talk.
There's an additional question.
Okay, thank you, Peter. So the question is, sorry.
Go ahead.
So the question is, how did you come up to build your library? Just a short history, if possible.
Yeah, very quickly, we built this library by contacting prominent practitioners in the field, because the monoterpene indole alkaloids are historical in a number of laboratories in Europe. So we have simply contacted them, and they accepted to share their history with us. And we started the LC-MS acquisition data, and we built the library. That's all.
Okay, thank you.
I have a quick question. So have you explored sort of chemoevolution in relationship to activities? And is that of interest based on you? Because these molecules are so beautiful, and now you have these networks. There may be trends that you may be able to spot.
I'm not sure that I have heard everything, but if you are talking about biological activity, we didn't try to map the biological activity on our networks for now. We are really focusing on the structural novelty. But after that, we share our compounds with labs for the biological evaluations.
Just a few words about the network. How does it work? The network you speak about.
Yeah, it's a little bit difficult to be brief about network, because it contains three steps. Briefly, networks allows to expand the annotation on a molecular network, starting from the in silico metabolization of a known compound in your network. So first, you have to identify a network on your compound on your network. Second, you will use a biochemical transformation to generate an in silico metabolite. The third step will be to perform the in silico fragmentation of the in silico generated metabolite to generate a CFMID MS-MS spectrum. And after that, there is a comparison between the MS-MS in silico spectrum of the in silico generated compound to the actual extract or experimental data. So it's not so easy to explain this very quickly, I'm sorry.
So would you potentially be interested at some point giving a workshop on network? I think this community would really benefit from that.
Yes, I think we can talk this with Gregory, which is the conceiver of this tool. I'm sure he will be really interested to do that. And I would be happy to show the examples.
Okay, do we have time for one question?
Yes.
Okay, so the question is from Thiago, and how do you map the genomic info to your molecular network, Mehdi?
For now, we didn't use this technology. We didn't use this in our work. I didn't map genomic information on network. I'm sorry.
Okay. So I think now we can go further, and again, I would like to warmly thank Mehdi for this talk. I think our next speaker is, I don't have the program on my screen,
Thank you.
is Joe Wandy.
Hi, let me share my screen now. All right. Okay.
So Joe, you have 8 minutes to present your work. And
Okay.
so,
Hi. Thank you all for. Sorry, can I start now? Yeah. Thank you all for coming to this talk. So my name is Joe Wandy from Glasgow University. So in my talk today, I'll be presenting our work on ranking metabolite sets by their activity levels, done with colleagues like Karen, Isabel, Justin, Simon, and Ronan. And I will also introduce a tool called PALS that would let you do that yourself. So going on, frequently when trying to make sense of complex metabolomics data, right, rather than trying to analyze individual metabolites in isolation, we would consider analyzing sets of related metabolites instead. So for example, we can group metabolites based on their participation in a series of chemical reactions forming metabolic pathways. Or a different way of producing metabolite sets is to group metabolites based on their fragmentation spectra similarities. So as you all know here, molecular networking can be used to group metabolites into molecular families, which could potentially correspond to novel chemical classes. And you can easily run this molecular networking analysis on top of GNPS. Another example of how metabolite sets can be created is demonstrated in our previous work of MS2LDA. So here metabolites are grouped by the co-occurring fragments and neutral losses using the LDA topic model into master motifs that could potentially correspond to chemical substructures. So you can easily run this workflow on top of GNPS as well. So a natural question that come up is to determine amongst all the list of metabolite sets that we get from an analysis, which one are interesting and should be pursued for further study. So there are many tools that could be used, for example, to analyze the enrichment activity of pathways. Keep in mind that a pathway is a specific instance of metabolite sets, right? But there are not as many tools out there that can be used to prioritize metabolite sets that come out from GNPS as well as MS2LDA. So to address this problem, we have developed PALS, an extensive and modular tools for metabolite sets prioritization. So PALS, the main objective of PALS is given a list of metabolite sets and the intensity values of the member peaks. We would like to obtain some kind of measure of statistical significance of those metabolite sets in different experimental conditions. So PALS can be easily integrated into pattern-based workflows and applications. Can be used to analyze different types of metabolite sets in forms of pathways. So you can pull all this pathway data from CAG and Reactome if you have it. You can also use PALS to import data from this feature-based molecular networking workflow and prioritize molecular families using it, or from a GNPS MS2LDA workflow and prioritize motifs. And the main output from PALS is a measure of significance in form of p-values on these metabolite sets. So this diagram here illustrates the overall schematic of how PALS work. On the left-hand side, panel A, so you see this example molecular family that comes from say GNPS FDMN analysis, right? So this includes the assignment of MS1 peaks to molecular families, as well as the MS1 peak intensities. We can, for each metabolite set, we can construct this matrix of intensities that you see on the right-hand side. So the rows are the MS1 peaks, and the columns are the different samples, which can be divided into, say, two groups, the case and the control. And To get an overall estimate of the activity levels of this metabolite set, so we use this procedure called PLAGE that has been used for analysis of gene expression level. From our observations, the method seems to generalize well to analyzing metabolite set as well. So in PLAGE, we first standardize the expression values by calculating the Z-score, and then we perform a singular value decomposition. The first right singular vector, so analogous to the first component in PCA that captures the largest variation in beta, is used as the activity level score. So given two experimental conditions, say the case and the control, we can perform a t-test to get the t-statistics between these two groups. And finally, we do permutation tests by permuting the sample labels to get the null distribution. And from here, we can obtain the p-values. So here, just more details on what I just described earlier. So using this method, we try to apply this on some example data from GNPS. So we demonstrate the use of pulse to analyze molecular family data using an American Gut project that was previously run through the feature-based molecular networking workflow from GNPS. So in this data, a subset of the full data was taken consisting of volunteers who are eating different amounts of plant-based food. The case group consists of people who are eating more than 30 plant-based food per week, whereas the control group are those who eat less than 10 plant-based food per week. So we use pulse and prioritize the molecular families that are found in this data. And overall, we found 35 molecular families with a significant p-values less than 0.05, who also have at least 10 or more member peaks in it that are significantly changing between the case and the control group. So we highlight here in particular, for example, is one molecular family that contains multiple hits to a steroid-related GNPS library hits, and it's found to be more abundant in the control group. So this could be explained maybe as a result of the abundance of steroids in meat, fish, and eggs as a result of the diet of the group. Other molecular families do not contain many GNPS library hits, but they could be an interesting avenue for further investigation. Another example, we take pulse and run them on this data set of 70 Rhamnaceae species from 2 clades and various genera. So this was previously run on the GNPS MS2LDA workflow. And from the original study, 25 master motifs potentially corresponding to novel chemical substructures were manually characterized in the original study. So using pulse, we found results which are consistent with the original study, but keep note that the previous efforts rely on manual interpretation, which could be time consuming. Whereas from pulse, we get the statistical significance of these master motifs or metabolite sets easily. And so some interesting results we found that were consistent with the original study. For example, pulse reveals that the master motifs annotated with the flavonoid-related substructures are differently expressed between the Rhamnus and the Ziziphus genera. And differential metabolite sets for other genera could also be examined in pulse. Just a click of a button. For example, the xylose or the arabinose moiety substructures was differentially expressed between Ventilago and Rhamnus. So if you want to try pulse yourself, you can visit our website, https://pulse.glasgowcombio.org. So pulse can be used from the command line or can be imported into Jupyter Notebook or any other Python workflow that you have. And there's also a nice web interface that you can use. So for example, if you like programming, so here it shows an example of a Jupyter Notebook that demonstrates how to run pulse. So at the top, you have to specify the URL to the GNPS analysis that you have done, a feature-based molecular networking one, and provide some kind of metadata in the data frame and the description of the case and the control group. Pass it to the loader objects, and then it will load the data and pass it to the PLAGE method that will then do the decomposition as well as finding the p-values of the molecular families. If you prefer to focus on the chemistry, we also provide this nice web interface that you can use to analyze different types of metabolite sets, including pathways, GNPS molecular families, or the MS2LDA analysis. So it's the same thing from here. You need to provide the URL to an existing GNPS analysis, as well as the metadata information. And if you click go, and then you get on the right-hand side a list of the metabolite sets that have been prioritized according to the p-values. And that'll be the end of my talk. Thank you, and if you to ask any question.
Thank you so much, Joe. We have a few minutes for questions, and there is a question. If you can comment on how PALS would perform compared to merging feature abundance at the subnetwork level and running standard statistic analysis.
Can you repeat the question again? How does PALS compare to
merging feature abundance at the subnetwork level and running standard statistics analysis?
So at the center of PALS, we do standard statistical analysis, right? We just do t-tests from the decomposition of from the singular value decomposition that we get. And at the moment is limited to comparing two groups, case and control group. And I guess the key thing is that if you just do standard statistics on a peak level, so it's like that gives you results on the peak, right? But we are interested to analyze a group of peaks, so to say, basically metabolite sets, which are collected together through some ways, either through knowledge based on pathways or through molecular networking or other MS2LD analysis or such things.
Thanks. There was another question, but I think someone has already posted. If there has...
Fidel, there is a question about, is there an R package that includes the PALS function?
Yeah, so sorry, the decomposition part, let me go back a few slides back. The decomposition part that we use inside PALS package is a place method. There is an R package, but I think on top of this decomposition, we also need a bunch of codes to retrieve the grouping itself. So say pathways from database or molecular families from GNPS and all that. So that, I don't think there's any R package to do that yet. However, it probably is not that hard to convert all these parts of the codes from Python to R. So this can be used in R as well.
Yeah, I think there's a lot of interest in using it in R, so that may be something to consider.
Yeah. So the main reason why we do this in Python is simply because in Glasgow Proteomics, we do a lot of work in Python. Right? I understand there's a lot of interest in using R as well from the community.
Fidel, can I use the microphone and ask a question?
Yes, Ricardo. Yes, go ahead.
Hi, Joey. Very nice tool. I'm just wondering, have you compared with like traditional methods for set enrichment, like hypergeometric test or?
Yeah, actually, so sorry to interrupt. So that's not included in this talk, but we do some comparison using synthetic data against ORA, for example, which uses hypergeometric, right, and GSEA as well. And I think our findings is that so the typical problem with metabolomics is that often the data is quite noisy, right? And then we get missing peaks or things which are not there, but it's actually not there. And when we try to simulate this kind of experiments in synthetic data, so we take some ground truth that we know and we progressively remove peaks. We found that this Plage method is really quite robust to noise. So it still performs well in finding significant things, even when there's a large number of things which are missing compared to, say, ORA or GSEA.
Great, thank you.
Yeah.
Thank you, Joe. I think we can move to the next speaker. It's Vicky, who will be talking about the use of GNPS to mine the fungal metabolome. Vicky, if you can share your screen, thank you.
Hello, I'm going to talk to you today about using GNPS to determine optimized strategies for continental scale mining of a fungal metabolome. And there we are. So many natural products discovery operations rely on libraries, whether they be microbial libraries, compound libraries, plant libraries, but the guidance behind building these libraries have been lacking. They sort of just rely on hand waving and common sense, sort of, but we hope to put some actual evidence-based science behind these strategies. And to do that, we're going to focus on a portion of our library that is housed at the Natural Products Discovery Group, which is a citizen science soil collection program, which is built of 53,000 fungal isolates that are isolated from soil samples that are sent from around the country from citizen scientists. This is a plot that shows the genetic diversity of our collection, and we have a lot of fungi. They come from nearly 500 genera, and we have a great breadth of Penicillium, Fusarium, Aspergillus, but we also have a lot of lesser known fungi that are represented by only a handful of isolates. And we wonder how our library composition, how effective it is, and how we can use this library to determine the most effective formulation for a library in the future. The goal of this project, therefore, was to study the chemical and genetic diversity encapsulated within a single fungal genus. And we did that by generating ITS sequences and a phylogeny based on those, and then generating and analyzing extracts run on LCMS-MS, and then analyzed by GNPS molecular networking, among other tools. We chose to focus on Alternaria for this project because it is a diverse fungal genus, it is chemically talented, and crucially, we had a nice sample size. And they are well distributed across the United States, so we thought we could pull in some geographical aspects as well. Now, looking at the most broad view of Alternaria, I'll draw your attention first to the feature accumulation curve on the left, and we can see that from this data, the chemistry of our Alternaria is well described through this analysis. On the x-axis, we have isolates, and then on the y-axis, we have features, and as we increase the number of isolates in our analysis, the new features also increase until we reach a plateau point up here somewhere. And we assessed the slope of the penultimate point, which is 1.42, which says that on average, if we added an additional isolate, we would only add 1.42 additional features. So we've got a pretty good handle on the chemistry of Alternaria. We also did our ITS phylogeny. This is the phylogram of our Alternaria, and based on this ITS sequence, our Alternaria can be divided into 5 distinct genetic clades. And I've numbered or lettered these U through Y, and the color scheme should be consistent throughout this talk. Clade U is the largest clade, made up of more than half of our samples, followed by V, W, Y, and X. And while we've got a pretty good handle on the chemistry of Alternaria as a whole, as a genus, we wondered what the chemistry of each clade looked like. So we did feature accumulation curves by clade. And we found that the larger two clades, clade U and to a lesser extent clade V, actually have a great deal of the chemistry already described. Their slopes are in the single digits, meaning that on average, fewer than 10 additional features would be added to the analysis given more isolates. It's a very different question when you come to the smaller clades, where we have double-digit slopes and they're a bit more variable, which is emphasized in this plot on the lower right, where we have the slope of each curve against the number of isolates. And when you have a lot of isolates, your slope is pretty low, your chemistry is well defined. But if you have only a couple of samples, fewer than 20 in this case, your chemistry you have a lot of room to grow and more chemical space to explore. So I think that the 416, I think, small genera in our library are a very interesting and profitable place for us to be exploring in future. I keep doing that. In addition to looking at individual chemical features, I also use GNPS molecular networking to do a network, and then I condensed these networks down into the chemical family level. So rather than looking at each individual node, I'm looking at this subnetwork or chemical family as a whole. And in contrast to the curves that I've shown you previously, you'll notice that these curves, they're also feature accumulation curves, but they don't have that beautiful smooth curve. These are collector's curves, which just puts the raw data right onto your curve. There's no modeling, there's no nothing. And using this type of curve is fun. You can strategically sort your data in a number of ways and find some very interesting answers to questions. In this case, I sorted the data clade U, V, W, X, and Y in the upper panel, and in the lower panel, I reversed that sorting. So you can say that if you have a whole lot of clade U, we had, I think, 111 isolates from clade U, 222 duplicates, you get a whole lot, most of the chemical families that are in Alternaria. And then by adding these additional smaller clades, you do add a couple more, a few hundred more chemical families, but you've got most of the way there using just clade U. Turning that on its head, you can also look at the opposite sort, where we start with the small clades that the chemistry was not well defined previously, but if you put them together, you actually arrive at the same number of chemical families without using clade U at all. Now, of course, once you add clade U back in, you do get that bump from clade U, and this is due to the number of unique chemical families as compared to the number of shared chemical families. And this is something that's very common in fungi, and I imagine other groups of organisms, that there is a lot of shared chemistry, so that if you are building a library, it may be possible to get a similar level of chemical diversity with fewer isolates if you emphasize smaller clades rather than just all of this one clade. Turning to geography, we wanted to know if there was a particular area of the country that had a higher chemical diversity of Alternaria. And we did see that there was an interesting distribution of Alternaria across the United States. For instance, clade U, our largest clade, is pretty much everywhere. And clades W in orange here and Y in purple are mostly constrained to the western portions of the country. But when we broke our data down by region, and again generated feature accumulation curves and plotted that against the number of isolates, we didn't find any truly diverse region in the country that went against our previous trend of just having more isolates gives you better diversity. So I would like to leave you with these final thoughts, that genetic diversity is important in library building, but it shouldn't be your only consideration. Redundant strains will still contribute to chemical diversity, and the best characterization of chemistry will be achieved with larger number of isolates. In this case, 40 isolates for Alternaria seemed good. It might be very different for other genera of fungi or other organisms altogether. Thirdly, a more efficient library may be possible if you build it based on a chemical family level rather than a chemical feature level. And finally, we did not find a hotspot where the perfect Alternaria was located. But broad sampling is beneficial to library diversity because you might have differential genetics within the sampling zone. Finally, I would like to thank everybody on this slide, my department and lab, everybody pictured, and especially I would like to thank the citizen scientists who submitted soil samples, because without them we wouldn't have a collection at all. So I would be happy to answer any questions if there are any.
Thank you so much.
Victoria.
So there is a question. Yes, I'm here. So there is a question from Scott, and he asks you, do you test multiple fermentation conditions to try to maximize your coverage of the chemical space?
We didn't do that. Oh, sorry, were you continuing? Sorry.
And how do you account for potential missing chemical diversity by not tracking silent gene cluster by alter fermentation conditions? So,
for this case, we wanted to just use a single fermentation condition so that we would have a very consistent data set. If we did try different conditions, of course, it would yield in different chemistry, but we wanted this first foray into this exploration to be very clean, and so we just chose the one condition for this project.
Okay, cool. So there is question about the use of, sorry, maybe Fidel, you want to ask something?
Yes. Yes, Victoria, the other question, I think it's similar to what you answered. The conditions.
Okay.
Was about the changing of different growth media.
Mm-hmm.
How that would affect the different clades chemically.
The clades would stay the same because those are based on ITS sequence, so genetics rather than chemistry. The changing media would change the chemical classification, so we would expect to see a different range of metabolites, but we haven't gotten a chance to do that yet.
Okay.
Gustavo asks you if this study is already published.
It is not. We're working on getting it out soon, though.
Okay. and thank you so much. I think maybe does the fungal morphology track with chemical diversity to layer in with genetic diversity?
There were different chemical or fungal morphologies that were observed on the plates. We grow all of our fungi on Cheerios, so I don't have fewer pictures, I suppose, of those. But there were, yes, they do have different morphologies based on genetics anyway, which is how I was sorting things originally. I could go back and check on the chemical relationship to morphology, but nothing stands out immediately to me.
Okay. Yeah, Mauricio asks you, the next factor is to explore the natural ecosystem where these fungi are present. By knowing the ecosystem, it would be possible to know what chemistry is needed or used by fungi to live and colonize environments. And he said, nice overview and work, and thank you for this very nice talk.
Yes, thank you. Yes, thank you.
So I think now we could move to the next speaker. Thank you so much, Victoria, for this work.
Thank you for listening.
Our next speaker is Yung Liu. The floor is yours.
Okay, thank you very much for giving me the opportunity to present our recent work on genomics and metabolomic analysis of potato common scale pathogen Streptomyces gibii. Sorry. So Streptomyces gibii is the causal agent of potato common scab disease. And this disease is characterized by the formation of a superficial or deep-pitted raised scab lesions on the tuber surface. And these lesions negatively impact the market value of potatoes, which causes significant loss to potato growers and worldwide. And Streptomyces gibii can infect a variety of plant species, include beet, turnip, and peanut. And it can produce several phytotoxic specialized metabolites, for example, sextomins, Coronafacoyl Phytotoxins, and concatamicins, and which are known or suspected to contribute to host pathogen interactions or disease development. However, the full potential of Streptomyces gibii has not been previously investigated. So the objective of this study is to using a combined metabolomics and genomics approach to investigate the metabolites that are produced by Streptomyces gibi. The genome sequence was analyzed using anti-smash and deep BGC to predict the biosynthetic gene clusters for specialized metabolites. And the Untitled LC-MS2 was used to characterize the metabolome of Streptomyces gibi and 3 culture conditions. And the MS2 data was analyzed using feature-based molecular networking and hierarchical clustering in Biodendron. And the metabolites were annotated using GNPS library search or by using NAP network series CFMD. So anti-smash predict 34 specialized metabolites biosynthetic gene clusters. The combined links for these BGCs accounts for 11.5% of the genome. And the deep BGC predict 146 specialized metabolites, and which makes up 25.03% of the genome. So the metabolomic analysis reveals that the metabolomic profile of Streptomyces gibi varies among 3 culture media tested, and with most metabolites being observed in the plant-based media OBA. And as the limited time, I will pick up only 2 metabolites that were detected in the metabolomics of Streptomyces gibi. And the first one I want to share with you is the sextomins. Sextomins are key pathogenicity determinants produced by Streptomyces gibi and other scab-causing Streptomyces species. And using feature-based molecular networking combined with Biodendron, multiple sextomin analogs were annotated. And the sextomin A were annotated using GNPS library search, and other analogs were annotated using either network CFMD or NAP. And Streptomyces gibi is also known to produce a trace amount of sextomin C and sextomin D, but initially sextomin C and D were not found in the main sextomin network created by feature-based molecular networking, whereas they can be detected in the Biodendron tree. When cross-checked with the feature-based network, sextomin C and D were found to form a separate cluster from the main sextomin cluster. On the other hand, some nodes in the Biodendron are not present in the feature-based molecular network. No, some nodes in the feature-based molecular network are not present in the Biodendron, and which appears to a limitation for Biodendron. That's why both feature-based molecular networking and Biodendron are both used in this study. And these two tools are complementary using different realization and similarity method. And 3 dehydrated sextomin A analogs were also putatively annotated using network, and these dehydrated analogs had never been reported before in Streptomyces gibi. Second, I would like to share the Coronafacoyl Phytotoxins with you. Coronafacoyl Phytotoxins are non-host specific, and though non-essential for the pathogenicity, and they contribute to the severity of disease symptoms induced by the producing organisms. And based on their precursor AMS, MS2 fragments, and CFND network predictions, 5 different Coronafacoyl phytotoxin analogs were putatively annotated, and the methyl substituted CFS lucin has never been reported in Streptomyces gibi before, and they are novel Coronafacoyl phytotoxins that differ from other phytotoxins in that they contain a methyl group in the C7 position of the bicyclic ring instead of an ethyl group. So other compounds such as conchylamycins, botulomycins were also putatively annotated in the Streptomyces metabolomics, such as conchylamycin, botulomycin, and compounds unknown to be produced by Streptomyces gibi were also putatively annotated from the 15 most intense areas, such as cyclo, L-valine, L-proline, aerogene. So in conclusion, we use a combined genomics and metabolomics approach to investigate the metabolites that are produced by Streptomyces gibi. And we showed that Streptomyces gibi genome contains a large number of biosynthetic gene clusters, including various of molecules with diverse bioactivities. And using untargeted metabolomics, non-Streptomyces gibi metabolites and putatively new analogs were annotated. And new molecules unknown to be produced by Streptomyces gibi were also detected and annotated. And overall, our study represents the first detailed analysis of the metabolome of Streptomyces gibi. Further research is required to characterize the predicted novel metabolites, identify and the associated biosynthetic gene clusters. And I would like to thank my supervisor, Dr. Don Bignell, and our lab group. Thanks Dr. Tlan and his lab member, Ashad, and thanks our collaborators from University of California, Dr. Nostes and Dr. Doristin. Thank you.
Thank you so much. You are perfectly in time, so perfect. We have time for some questions. Maybe Peter suggests that there is enzymatic promiscuity and the molecular networking can reveal this promiscuity. What do you think about that? Maybe Peter could comment.
Sure. So one of the things that I saw, you had the alpha group versus a methyl group.
Yes.
From one of your structures. Do you think that is because of catalytic promiscuity of the enzymes?
Yes. And based on our previous studies, and we putatively found this molecule, but we are not sure. Then this time we use CFM ID, and based on their MS2 fragmentations, and we compare and using CFM ID and found this one is the best candidate, the methyl substituted.
So we have also a question from Louis Felix that asks you to elaborate more on the reason why there is different number of gene annotation obtained with anti-SMASH or deep BCG.
Yes, they use anti-SMASH and deep BGC using different methods to predict the biosynthetic gene clusters, and both of them have their own advantages and limitations. Because anti-SMASH using a machine learning method, the hidden molecule models, and a human-defined rules-based approach to identify biosynthetic gene clusters. And the deep BGC using a deep learning it's a deep learning approach, and using the recurrent neural networks and the pump domains to predict the biosynthetic gene cluster, and which is excel detecting as novel biosynthetic gene clusters.
Okay, and what about the difference, the similarity measures used in Biodendro?
Yes, both the feature-based molecular networking and Biodendro, they process the data similarly, like they both process the MS2 data with the MZ-Man2, and then feature-based molecular networking, yeah, they both process the data similar, but then they submit they just using different forms and submit to their systems, and the Biodendro one, the Biodendro one is it's using dynamic binding method, and the features were aligned to their respective MS spectra. And all the product areas were ordered by MZ and a new V, and was created when the difference between two consecutive masses exceed a user-defined threshold. And some, yeah, I think, and also the pairwise distance, the Bell-Dendra using the Curtis method or the Dickert method. I don't know the feature-based one using which one, but I think it's similar.
Okay. Okay, thank you so much, Liu. I think just a last question from Scott Jarmusch that asks you if you use NMR to confirm the new structure that you get
No.
with your no?
No, we haven't done that yet. No, we just putatively annotated them, and we'll do that.
Okay. So thank you so much for this nice work and nice presentation. I think now it's time for coffee, and we have the coffee break. So I think please come back before 10:30, and then this is the end. Maybe we have time for conclusion, sorry. If some of you want to have a brief conclusion, I would like to warmly thank all these presenters, the keynote speaker, and all the short talk. That was very interesting. And as Peter said, the community is participate and as all the speakers show, we have some community that brings some data, some mass spec data, and that's very interesting for GNPS and for molecular networking. So now it's time for coffee break. So come back before 10:30.
Thanks a lot, Audrey, and thanks a lot to all the speakers. That was awesome. Before you guys go and grab coffee or tea or whatever beverages and food, I just wanted to remind you that we also made a meeting survey. So if you haven't filled it out yet, I posted a link in the chat and I will repost it in a second. So for us to get feedback, that would be actually really valuable. So yeah, if you can spend 2 minutes of your break to fill this out, this would be awesome. Thanks.
Hey everybody, it's time to move forward. My name is Ricardo. I'm a former member of the Ornstein Lab, and I'm now a researcher at the University of São Paulo, where I lead the computational chemical biology lab. And for me, it's very nice to be participating of this first GNPS user meeting. I've been following GNPS since 2015, and it's great to see how it's getting like the community together. It's really great, and I'll be sharing the natural product section with Luis. And Luis, would you like to introduce yourself?
Yes, thank you, Ricardo. Hi, everyone. I'm Louis Felix, and indeed, I will be co-chairing the network session. And I just wanted to thank the participants and the speakers for their presentation today. I'm looking forward. Thank you very much.
Okay, Louis will be helping me mainly with the questions, so we'll be going back and forth. And it's also a pleasure to me to welcome Professor Carrie McPhail from Oregon State University. I met Carrie briefly at the Dornstein Lab, and it was always very nice to discuss science with her. I'm looking forward for her presentation entitled GNPS Molecular Networking Tools Guide Biological Activity Hypothesis for Complex Extracts. Carrie has been using molecular networking to study a variety of interesting biological systems, including fungi, marine organisms, and also South African stromatolites. Thank you, Carrie. You have the microphone.
Carrie, you need to unmute yourself.
No, check to see if your microphone is plugged in or your headphone. We tested it earlier with her and it was working, but it's not working at the moment.
Yeah, just a second.
No.
Yeah, we can't hear you now. You are unmuted, so it should be on your computer.
Is your headphone plugged in?
Yes.
Ah, now we can hear you. Say something.
All right, so can you hear me now?
Perfect.
You can hear me now?
Yes, we can.
Right. Wow, I'm sorry about that. As they said, it was working just fine before. So
don't worry about it.
Let me just share my screen again.
We expected hiccups and we budgeted time for this. So
and
you'll have your full time.
There we go. All right, so I'm sorry about that. So thank you to the Darstein Lab organizers for this great opportunity to share a little of how we use molecular networking tools, where our goal is to find biologically active natural products with relevance to human health and agriculture, really relying on the premise that this great biological diversity equates with chemical diversity. So our focus has been primarily on cytotoxins as potential anti-cancer leads. Here are some of the compounds discovered or rediscovered in my lab that we continue to work on for the mechanism of action in collaboration with the Ishmael lab. Notably, these natural products were discovered through laborious reductionist approaches of traditional biological acid-guided fractionation, and we didn't learn much about the producing organisms in the context of their ecology and evolutionary history, which can provide important clues to the biological function and supply. So today I'm going to tell you though about living modern stromatolites as opposed to the ancient fossils that you may think of when hearing the term stromatolite. There's been much debate about how these form and their role in the global carbon cycle, among other things. Why do these microbial communities accrete calcium carbonate and phosphate when free-living microbial mats do not? On the right are electron micrographs that really show the mineralized microbial sheets forming hollow tunnel structures, and then also sheets of exopolysaccharide that bind the cells in the matrix. Importantly, this backscattered image here, paired with calcium element and phosphorus element distribution maps, shows that the calcium carbonate in pink in high abundance here is deeper in the stromatolite core at right, and then really the phosphate is higher at the surface, decreasing left. So it forms a phosphate crust above the carbonate. And this is very similar to the fossil record. They're not just microbial accretions of carbonate. So in thinking about this, we decided to look into the chemical signaling of these very distinct South African stromatolite microbial consortia, essentially asking whether we could put microorganisms and even their secondary metabolites into kind of functional guilds. And here we have a high resolution GIS map of the specific study site called Schoenmaker's Cup pool, and the colored squares with numbers represent collection sites around the pool that are labeled with numbered flags. So substrate cores were taken in quadruplicate, we call them tissue or substrate cores, at 3 levels immediately around each flag, as you can see here. And this work was actually done by Emily Gentry, Allegra Aaron, and Daniel Petrus, who came with us in November 2018 to South Africa. So we also at the same time took water that was collected at each flagged point as possible to analyze dissolved organic matter following the protocol described by Daniel in 2017 for the analysis of DOM. And just to show you the broad workflow, material was collected then for chemical extraction, ultimately serving for some chemical and biological characterization of metabolites, while DNA extraction really allows for metagenomics work led by the Quan lab, directed towards characterization of organisms, the primary mode of metabolism, and the secondary metabolite biosynthetic gene cluster predictions, using tools like anti-smash, for example. The purpose of this slide is really just to acknowledge the broad scope and large number of people involved, and to say that the focus today is on a very small part of the project. And I just want to first say that I was also curious, where do these data fit in with the broader community data for GNPS? And I thought to use Redo out of interest. So just quickly, here are the stromatolite samples in dark blue. And in fact, the core tissue samples are in the lower right here, while the DOM samples actually cluster towards the upper right. And this fits, this pale green dissolved organic matter water saline is more towards the upper, while in the tissue core samples better overlap with the gray cultured bacterial. So this was great to see the logic using Redo, and I think that's a great fun tool that you can get lost in there. But so what did we do? Well, we used biological screening in parallel cancer cell viability and mechanism-based assays to screen a small but bulk sample sent to us, which comprised a fistful of material combined from flag sites 1, 3, 7, and 10 here. And these were collected earlier in March 2018. We have to be mindful of preserving the sites, that we started with the small sample. This extract and its first tier fractions were tested against these engineered glioblastoma brain cancer cells that secrete fluorescent Gaussian luciferase protein. So loss of fluorescence indicates inhibition of protein secretion. What we want to see after 16 hours is still good cell viability, as shown by these orange dots, but low Gaussian luciferase secretion, similar to control aprotoxin here. Make no mistake, these cells will die by 48 hours. So here, this BCI33H looked most interesting, and George Neuhaus took a look at that. In order to digest still these rather complex LCMS data for the BCI33 fractions, GNH even, George used ion identity feature-based molecular networking to really simplify the network, even for these few fractions. And what stood out to him straight away, he honed in on this subnetwork because it looked like a cluster of peptides. And he knows that my lab has been interested in these as inhibitors of protein secretion, particularly. So here we can see this mass spectrum below, in particular, just the loss of 147 for phenylalanine, and also this carbon oxygen CO loss is very distinctive here, but overall looked like a peptide. And why would this be important? Well, on the right are all the natural products known, currently mostly cyclic peptide, that inhibit protein secretion by blocking the Sec61 translocan pore through the endoplasmic reticulum membrane, where all newly translated peptides actually enter the ER to be processed for secretion outside of the cell or insertion into the membranes. And this is an important process because membrane and secreted proteins account for 30% of the human proteome. And importantly, there's several targets of protein secretion along this pathway. Not all inhibitors would bind to the translocan at the entry to the pathway. Notably as well, viral packaging proteins, including coronavirus spike protein, is processed through this pathway. So anyway, if we look at the biological activity overlaid on this network for these BCI33 fractions, we see that peptides occur in fractions with potent activity, less than 40% Gaussian luciferase secretion, but also in fractions with minimal activity or even none at all. So George turned to, this was a bit disappointing, but there were certainly some other clusters that did have some potently toxic compounds. And he found that the MSMS spectra were not all that great in positive mode, so he created a network of ESI negative data. And looking at this simpler negative mode data with biological activity overlaid on the network, he could see that some of these nodes only occurred in the most active fractions. And so what was this? Well, there was also, these were annotated with this bronze library hit of a glycosylated acyl glycerol. signaling that it had a similar MS2 spectrum, even if the parent mass was different. So what was this? Well, actually, George used NP-Atlas, the Linnington group tool, and really came up with this very nice match for the 491 mass here. Remember, this is M-H and the formate adduct, and this is from a freshwater cyanobacterium, Oscillatoria rosea, so completely consistent with where we're looking. And it is always important, you know, to go back to the raw data. So here is one example of several related peaks in the TIC, showing the MS2 fragmentation pattern, really typical for a sugar as well as the acyl chain. And so we also knew, right, that there were other clusters. And what are these? Well, very similar. If we look at these other bioactive subnetworks, we see that they relate to disaccharides. And they actually didn't cluster together in feature-based molecular networking, which may not be surprising. So George switched to the classical molecular networking, which would then condense these. And you can see a very nice cluster together of the monosaccharide and disaccharide. And obviously, we want to have these kinds of compounds as standards for our assays so that we can potentially identify them from their specific profile in the assays for future dereplication. And so we would need to HPLC purify them and confirm identity now. But are they the only actives? Well, the answer is no, because while we were talking about the BCI33H activity that we showed, George had performed another level of fractionation. So the glycosylated acylglycerols I just told you about were actually from subfraction H3, while in fact H1 and H2 were also active. So the story continues. But you know, we'd been working with a bulk sample from some combined sites collected at an earlier date. So we really wanted to know how the 3 or 4 sets of compounds that we focused on so far were distributed across the stromatolite pool and how important. So thanks to Allegra, Emily, and also Kelly Weldon, this Bray-Curtis analysis showed the relationship of different flag sites and notably some primary drivers. I don't want to discuss the hypothesis of what accounts for this particular pattern of the metabolites here, but I do want to draw your attention to this list of the features that are drivers for this separation, in particular 2198. So this is a global network of all 131 stromatolite tissue cores and was constructed using feature-based molecular networking of the ESI positive data. And feature 2198 here falls in this really large cluster. So this was actually exciting for us because it had the same mass as the peptides that George was looking at earlier. Is this peptide or similar ones found in any other samples with data submitted to GNPS? Well, we used MAST for that. And in fact, doing MAST or the single spectrum search says no, the only data sets it's found in are actually the stromatolite DOM, and then the stromatolite tissues from November and also the earlier March collection. So we couldn't say that these had been submitted previously, but then using the in silico dereplicated tool, you know, very crudely, this yielded only some very low scoring hits, right? A score of 7, but it's still useful information about substructure motifs. And if we go back to view a part of the subnetwork in the GNPS viewer, we get this directional edges, which I only saw recently and find this really interesting. And overlaying the delta MZ values on the edges suggests these losses of these amino acids and C6H12. So now all of these are really connected to this bigger mass here, H61. And if we do a dereplicated search on this feature now, we see that there's a hit for nostophysine. And again, it has this loss of CO, loss of CO. This is about as far as we've gotten. Nostophysine has a much larger mass at 888 Dalton. It's also related to a very large group of microcystins. So the next steps are obviously to investigate this difference and go back to looking at annotations across the subnetwork, perhaps to use all these great tools that we just heard about, MS2LDA and PELS. So with that, I'll finish by saying there's obviously a lot to do here, not just in analyzing this classical molecular network of combined tissue and DOM stromatolite metabolite samples now, but also combining those with the genetic sequence data. And I want to thank all of the people that I've mentioned, plus several more of our collaborators, as well as the Moore Foundation for funding. I know that Xavier Siwe Nondo is on the line here as well. And I want to thank you for your time. I would welcome any questions now or later.
Thank you very much, Gary, for your talk. That was brilliant. And we have several questions here. The first one is a question from Osama.
I'm not sure if anybody has any questions. Oh.
Yeah, about how did you...
we have used NMR to confirm the identity of our compounds. So I didn't show that. We have got spectra of the acylglycerols. We don't have spectra yet of the peptides. Obviously, we will do that work.
Okay, Carrie, can you hear me?
Carrie, can you hear Louis?
Okay.
No, so he's not.
So I can't hear anybody else. I'm sorry about that.
Okay.
Maybe we can move forward, and then at the end we ask the questions for her.
Carrie, I can hear you now. My apologies. I can hear you now.
Okay.
Great. Okay. All right. So there were several questions. One of them. So first, thank you very much for your talk. That was great, and there are several questions. The first one is, which tool did you use to investigate the relationship between bioactivity and the molecular network?
So we really just grouped in this case, we just did a simple overlay of the biological activity on the molecular network. So it depended, we knew which fractions were active, and we used that to overlay.
Got it. And that was using the feature-based molecular network.
Yes, yeah. Sorry. So we use the feature-based molecular networking also with the ion identity network, which we just find collapses those features very nicely and makes fewer clusters, obviously.
Right. And there is another question from Mehdi about the doubly charged ions. And I think that relates to the ion identity molecular networking, because in the network you showed, there are nodes for ions that didn't have an MS2 spectra, right?
Yes.
Okay, and you were able to aggregate these ions using ion identity networking.
Yes, so, with these peptides, we often get the doubly charged ions. A lot of them do give the doubly charged ions.
Yes. And another question from Mehdi about the extraction protocol for the stromatolite extract.
We try and keep it really, really simple. In this case, we just simply used methanol. We normally would use 2:1 dichloromethane methanol for cyanobacteria. So for the mass spectrometry, we just really tried to keep it simple. And we did some tests beforehand and found that there's really not any discernible difference.
All right, thank you very much.
Thank you. Sorry about the trouble with the headset.
It's okay, there's another question.
Peter, I think we'll forward the questions to the end now, just
okay.
So we can move forward. Sorry, Carrie. Thank you, Carrie, for your presentation. It was really nice. For our next short talk, we have Adriano Rutz from the University of Geneva. His presentation is entitled Taxonomically Informed Scoring Enhances Confidence in Natural Products Annotation. Adriano, whenever you're ready, you can share your screen and open your microphone. Thank you.
So can you see the screen? Can you hear me?
Yes.
Perfect. So today I'm going to present you a work we published last year, Taxonomically Informed Scoring Enhances Confidence in Natural Product Annotation. So first, natural product annotation. It starts with a natural product or mixture of natural products which we want to study. One of the ways to study them is to analyze their MS2 spectra and ideally to annotate some structures. But what makes natural products different from other structures? They're producing organisms. In fact, producing natural products are synthesized by living organisms. So this already focuses the possibilities on biosynthetically available molecules only. And the producing organisms can also be studied by botanists, mycologists, and so on to try defining them thanks to their morphological traits. And in between, there is the genetic material. It governs the ability of an organism to produce a given natural product. However, for given organisms like plants, DNA sequencing is challenging because their sequences are extremely diverse. So let's go back to chemistry. Once we annotated our spectra with structures, we can organize them in a chemical space. There are already multiple tools allowing to organize structures and spectra such as molecular network. When we talk about assembly and alignment of DNA sequences, we talk about phylogeny. And here again, tools such as MIBIG allow to make links between biosynthetic clusters and structures. It is currently used for microorganisms because they are quite easy to sequence, but non-sequenced organisms remain a challenge. So for living organism, according to their morphological trait, there is taxonomy. Benefiting from already established classification, it allows, in a certain way, to skip sequencing. And this is where our work takes place, linking natural products with their producing organisms and taxonomy. This is very powerful because the defined organism carries already all the genetic information itself. In a certain way, it's a shortcut between chemistry and biology without sequencing again. So let's see how this shortcut takes place in our scoring system. This is what most mass spectrometry users are used to, a score based on spectral similarity between a given spectrum and a candidate. It can be an in silico spectrum or an experimental one like in GNPS. But in most of the cases, this score is insufficient to annotate your spectrum properly. Therefore, people started to think about other scores to complement it. Brilliant idea was to give a score based on the structural consistency of your annotations. And this was, for example, done with the network annotation propagation or in Zodiac, a new tool available in series. Another parameter that can be useful to refine your score is physicochemical consistency. Tools like MedFrag, retention order prediction use it, and in gas chromatography, the Kovacs indices are used that way from many years. And now our proposal, the taxonomically informed scoring. Basically, it compares the producing organism of your spectrum and the one of your candidate and gives the score inversely proportional to their distance. Of course, once such ideal scoring system will be in place, there will remain the challenge of attributing weights to each of the scores. But this is not for today, and for today I will focus about the two first terms of our equation, since they are the one we benchmarked. Let me give you an idea about how our candidate re-ranking works. So first, you start with an unknown molecule or a mixture of unknowns. You acquire some MS2 spectra, and then you add the producing organism of your sample. More importantly, you can use any metabolite annotation tool you want. You do not need to change your existing pipeline. After your normal annotation process, you end up with a feature list with candidates that you complemented with your biological source. Once done, you re-rank your candidates according to the distance between the producing organism and the one of your sample. Here, for example, the spectrum initially ranked 6th is belonging to the exact same species of your sample. The one that was ranked first is belonging to the same genus, some other to the same families, or completely unrelated organisms. To summarize, what you need to perform your re-ranking, additionally to your spectra, are A, the producing organism of your sample, and B, a list of structures produced by given organisms. And this is what we wanted to build for our benchmarking dataset, a list of spectra, structure, organisms. So therefore, we gathered all the spectra and structures available within GNPS. We cleaned them to be sure to start with a good set, which still led us to more than 40,000 spectra structure pairs. We complemented them with the information coming from the Dictionary of Natural Products. Again, we cleaned all structures and organisms to be sure to have a consistent naming and taxonomy. And this finally led us to a benchmarking set of more than 2,000 entries with spectrum, structure, organism. So for the benchmark, we chose 3 state-of-the-art tools, namely Sirius, MS Finder, and our in-lab tool, ISDB DNP. And as you can see on the left, the F1 score before taxonomically informed scoring. To make it simple, the F1 score is an approximation of the correct candidates at rank 1. And no matter the tool used, correct structures ranked first were increased by a factor ranging from 1.5 to 7-fold. So ultimately, on our set, we reached more than 80% of correctly annotated structures at rank 1. We were also interested in the complementarity between the tools, and we saw that the correct results were not necessarily the same, showing the interest to use multiple approaches and to inform them taxonomically. So as just shown, taxonomically informed scoring is effective, but there is a strong requirement, which is the availability of structure organism databases. And even if we have wonderfully furnished organism databases such as GBIF with millions of entries, or on the chemical side, PubChem with hundreds of millions, there is sadly only very few databases linking organisms and structures, and all of them got some little drawbacks. So this leads me to the projects we are currently working on, which is the establishment of an open curated natural product database. We'll be soon populating Wikidata with our entries, and I'll show you briefly how. So this is a typical page of natural product in Wikidata, and it is described as chemical compound, like any other chemical compound. But what we are interested in is the natural product of taxon, and this is the precise field we will be adding. So this is the link between our structure, its producing organism, and the original article describing this link. This data will be open to everyone, accessible for curation, and will allow applications in many other projects. We currently have more than 150,000 of those entries, and in close collaboration with curation experts of Wikidata, we'll be uploading them carefully in the coming days. Now, some quick acknowledgements for the team behind these projects. Ladies first, Miwa Kubo, which initiated the taxonomically informed scoring. Jonathan Bisson, in collaboration from Chicago, for all the ontological knowledge he brought to us. Pierre-Marie Allard, last author of the paper, always pushing to improve our research. And of course, Jean-Luc Wolfender, my PI. And we have a brand new building, so if you have the chance to pass by or to simply contact us, just feel free. Thank you.
Thank you very much, Adriano, for your presentation. And we have time for a few questions. The first one from Guillermo about how do you propagate errors when using your scoring system that has multiple variables?
I'm not sure to understand which error does it.
So you have 4 variables in your scoring that you presented. And how do you account for errors for one of them in the scoring?
So basically the work we did was only on 2 variables of the 4, but of course the score is not perfect and will be developed to account for errors or other weights or other possibilities.
Okay, and another question from Rachel about she indicates the taxonomy can sometimes be misleading, and how is likely that the same compound is produced by two very dissimilar species?
It is indeed possible, and that's something we want to how can I say, to mitigate with our open database. So having more information, we will be able to see those strange compounds that are present in very different taxonomies.
Okay, so there are more questions and we will continue after. I have one last question before we move on. And yeah, I have the impression that if, like in metabolomics analysis, we detect a lot of compounds, but that scoring approach is efficient only if we have information between the species and the structure. And I'm wondering, like, would you recommend that method to be used on well-characterized species and not to be used for the species that are poorly investigated?
No, I would recommend to use it for all samples because the species that are poorly investigated will benefit from the compounds reported in the most studied one. So if you are working on whatever Asteraceae, which is poorly studied, it will benefit from all the compounds from the studied Asteraceae belonging to the same family.
Okay, but with the consequences that it's gonna bias the annotation toward the known molecules, right? Unknown molecules will be weighted down in the scoring.
Of course, but then if you have a cluster which is highly consistent to be unknown, it's even more consistent.
All right, thank you very much, Adriano. We'll continue later.
You're welcome.
Thank you, Adriano. Very nice presentation. Our next speaker is Scott Yarmush, affiliated to University of Aberdeen, Scotland, and Uppsala University, Sweden. His presentation is entitled Unraveling the Sphereoxamine Chemical Ecology Between Extremotolerant Streptomyces SPS 29 and Fungal Phytopathogens. Thank you, Scott. You have the microphone.
Yeah, thank you. So this is a really nice example of using molecular networking and some of these tools in GNPS to examine the chemical ecology of some of these species that a lot of the people work around the world on. So the idea in the Marine Biodiscovery Center, where I did my PhD, is that the extreme climates and extreme biospheres of the Earth, they make the organisms that live within them adapt strange biological adaptations to survive. And the hope is that this biological adaptation leads to interesting chemistry. And over the years, we've shown this in Marcel Jasper's lab in Aberdeen. So in this case, this was really started by a PhD student in Chile, Diego Lagos. And so he went to the San Pedro de Atacama in the Atacama Desert in Chile. And at this altitude, there are these lupine plants that bloom when there's enough water dumped over the Andes. And so the idea was that you have this lupine plant, you sample around the rhizosphere of the plant, the soil around the roots. And he identified over 20 Streptomyces strains that showed antifungal activity. So in this case, we're looking for antifungal producing Streptomyces species, which could be used for viticulture purposes, so for the wine industry. And so here's an example. This is how he screens the plates. So you have the Streptomyces species on the left. And now he's screening against Botrytis cinerea, which is a common wine growing plant phytopathogen. And so without explaining it too much, we translated this into the lab in liquid culture. We ran all the bioactivity guided fractionation. And we found that we didn't see any antifungal compounds, which is not unheard of, going from solid culture to liquid culture. But when we cultured the ending fermentation, we saw no growth of fungi. So we knew that the Streptomyces had implemented a way to prevent the growth of the fungus, but without antifungals. So what was this? So at the end of the day, it turns out they are these siderophores, which I'll show in a little bit, but essentially we have to first go through some of the biosynthesis. So this is a lot of work done in Greg Chalice's lab at Warwick. So here's a Streptomyces plate, and there's metabolite pools within Streptomyces and within any bacteria. And then you have a very basic gene cluster here. So it's consisting of 4 genes. And what happens is these metabolite pools feed these genes, essentially. Or this is the running hypothesis. So for instance, in the first gene, you have lysine that can be fed in, and that produces cadaverine. But in the lack of lysine, we can bypass this gene, but if there's putrescine in the environment. The most important gene here is this DESC, which typically is not a very promiscuous enzyme. It doesn't take a lot of different substrates, but there's been a few cases in Streptomyces showing that it can be quite promiscuous. And so it can mine these organic acids from metabolite pools. And these type of metabolites form in oligomers, and then they connect to each other. And so we have a lot of different desferoxamine scaffolds that can be produced. And a lot have been described so far. And then there's transport, and the point of these molecules is they uptake iron for the organism. So they chelate iron, and then they uptake them with a different protein. And so, of course, why we're here, we use GNPS, molecular networking, to visualize this. So this is the overlay of the network. And so essentially what has happened is we've grown the Streptomyces with the fungus and seen this action. So we have all the extracts here of the Streptomyces grown with different fungi, as well as the Streptomyces alone, and some HPLC fractions. And essentially, obviously what to focus on here are the molecular families that are circled. These all contain desferoxamine structures. So now we have to get into annotation. So here's our molecular family. We take a node from this molecular family, right? And we do what Mehdi described and also what Carrie described. So we predict a molecular formula in this case. We choose the one that's most likely. We look at the fragmentation pattern to see that it matches what we expect. And we start forming up structures. We can do this with these types of molecules because they are peptides in nature. So we can predict their fragmentation patterns with pretty high accuracy. And then, like any natural products group or structural elucidation group, we form the structure with the base structure, and then whatever is left, we can start coming up with substructure possibilities. Then we have a predicted structure on the left here. We do annotation, so we check back with the MS2 data to confirm and see if this is correct. So we went through this, and so here, all the nodes that are annotated are known desferoxamines. All the ones that are hexagons were in the GNPS library. And then all the nodes that have asterisks are new derivatives. So this is used using the connections within GNPS and also the molecular formula. And so what type of structural diversity do we see? So we see aryl desferoxamines. These have been described a few times. So some unique ones we have are this aryl desferoxamine with the putrescine molecule on the N-terminus. We also see the second example of a glycosylated desferoxamine. And then we also have acyl desferoxamines, which are more common. The first, we see the first derivatives that have this acetyl group on the end. And then we also see some of these monounsaturated derivatives. So through the co-cultivation, does this actually do anything? Or are we just seeing what Streptomyces can do no matter what? So in Streptomyces alone, the monoculture, we have 15 new analogs, 9 known ones. And in the co-culture, we have an additional 9 new analogs and an additional 15 known analogs. And the majority of these analogs here are acyl desferoxamines, which Matt Traxler showed that they are produced exclusively when cultured with other actinobacteria. So maybe we're seeing the same phenomenon with these fungal phytopathogens to try to fight them off. So essentially, what's happening is the organism is creating these siderophores to uptake all the iron so that it starves the fungus of the iron. So I said to myself, we wanted to look at what the chemical space looked like. And are we actually looking at everything that we have, or is there a lot of other desferoxamine potential out there? So you use MS2LDA, and Joe thankfully did a nice description of this. So here I've highlighted all the nodes that I pulled out from the normal molecular networking workflow. And when we run MS2LDA and overlap them, now we see almost triple the amount of nodes with double the amount of connections between them. So clearly there's a lot of other desferoxamine chemical space that is out there to be explored. So overall, we have the streptomyces, which showed a great potential for being antifungal. Using GNPS, we found a lot of desferoxamine analogs, MS2LDA showing we have a lot of potential. But really what it is that this streptomyces is using essentially a shield of desferoxamines to protect the plants, which could be used as a biocontrol agent. So just to thank the group in Aberdeen, MT, who was the master student who did a lot of the work, and the group in Chile, and Diego's, who is the PhD student there. And thank you.
Thank you very much, Scott. I think we have time for one question. And it's fascinating that you were able to expand your annotation in such a manner compared to just using the GNPS spectral libraries, or I don't know if you use the replicator in your study, but you were able to expand and propagate this annotation both manually by looking at the fragments and using MS2LDA. And I would like to know from a practical standpoint, how did you go back and forth between the molecular network and MS2LDA?
Yeah, so it's a great question. So really we're limiting our knowledge based on the biosynthesis. So because these are difficult metabolites to isolate and run NMR, because there's a lot of similar features in the molecules, it's tough to pick out unique features and sometimes. We use the known biosynthesis to do a lot of the annotations. And then MS2LDA pulls out a lot of things that we have no idea what could be added to the molecule. We can make hypotheses based on acids that could be potentially in metabolite pools, but this is kind of a dangerous thing without actually confirming the structure in any other way. So MS2LDA at the moment is being used as a novelty determinant. And it's very difficult to annotate structures using MS2LDA for these types of molecules, I think.
Thank you very much. I don't know, Ricardo, if we have enough time to ask more questions.
Can I ask a quick one, Louis?
Yeah, we have a lot of very interesting questions. We are a little bit over time, but I don't know. It's up to you.
I would go ahead and ask questions, a couple more. I think it's worth our time. We have 8 minutes afterwards, so.
Daniel, did you want to comment?
Yeah. Thanks a lot, Scott. That was really cool. I was wondering, like at the very beginning, you mentioned that the system you study is like an extreme environment, but so like the plant rhizosphere does not like necessarily seem super extreme to me. But what's the story behind that?
Yeah. So when the soil is taken and the microbes are cultured, we're only talking about maybe a few centimeters into the soil. So all of these microbes are still exposed to the high temperature, high salinity, low rainfall, and high UV radiation. And so we're not going deep enough in the soil for those microbes to be protected. But essentially, the microbes in the environment are dormant the majority of the time, which is what's been proven. But Mike Goodfellow's lab in Newcastle has shown that these soils, although they have common bacteria associated to all soils, like streptomyces and some bacilli, the actual microbial load in these soils is extremely low. So maybe 1 gram of soil in the Atacama Desert only contains 100 microbes, whereas 1 gram of soil outside contains, 10 million. So it seems that there are some microbes that are required no matter what type of soil we're in.
Cool. Interesting.
Louis, you want to?
Maybe one last question from Laura about, did you observe any of these compounds bound to a metal?
Yeah, so I just didn't include it in this because I didn't have time. So we saw a large molecular family that was chelated with both iron and aluminum. And so unfortunately, a side effect of these molecules is that they seem to chelate aluminum equally to iron, but aluminum is not used by the bacteria in any way. It's actually toxic to it. So it seems that it's a side effect of the biological activity of the metabolites. But yep, we absolutely do.
Got it. Thank you very much.
Thank you.
So our last speaker for this session before the discussion is Rafael Herr from Center of Marine Biotechnology and Biomedicine at University of California, San Diego. His presentation is entitled Deep Learning Guided Functional Metabolomics Enables Accelerated Natural Products Discovery. Thank you, Rafael. You have the microphone.
Right. Thank you very much, Ricardo. So when I first start working on a new crude natural product extract, I always feel both fascinated and overwhelmed by the sheer number of natural products and the pleiotropic bioactivities they might offer. So it's a great pleasure for me to introduce you today to a machine learning informed high throughput screening approach that leads us or helps us to target a new goldfish species out of a school of thousands of natural product fish in a crude natural product extract. So currently there are several bottlenecks that slow down the drug discovery process. And besides the problem of how to catch the fish, another major bottleneck is the time and cost intensive determination of the correct chemical structure of a bioactive molecule. We therefore envisioned an automated tool that helps us with the interpretation of a new molecule via the help of machine learning, based on only one very fast NMR experiment called HSQC. And this interdisciplinary effort yielded a tool that we call Small Molecule Accurate Recognition Technology, or in short, SMART. It consists of a convolutional neural network that was originally trained on experimental HSQC NMR data of 25,000 natural products. And since its development in December 2019, the tool has gained more than 1,000 users from all over the world, more than 50 countries, and serves them as a tool for faster replication of known and accelerated structure elucidation of new natural products. But to elucidate natural products, you have to first collect the producing organisms first. So we went on a sample collection trip to Puerto Rico with the Neyman and Caro Diaz labs, and where we collected, among others, a button-like cyanobacterium associated with brain cords. And then back in lab, it was identified as Rivularia species via microscopic as well as 16S rRNA analysis. And then we fractionated the crude extract via solid phase extraction into 4 subfractions and analyzed them for cytotoxicity. And we found the crude extract in fraction 2 to be highly cytotoxic, while the other fractions did not show cytotoxicity. And because cytotoxicity can be caused by many different mechanisms of action, we decided to have a look on the non-cytotoxic fraction 1 first to see if we can find hints for other more selective bioactivities. So we recorded an HSQC NMR spectrum of fraction 1, and subsequently SMART2 analysis suggested many chemically related peptides of the family of so-called HP cyclodepsipeptides, all of which are known serine protease inhibitors. And here highlighted is the name-giving 3-amino-6-hydroxy-2-piperidone unit, or short AHP. And you can further explore your SMART results in the Moliverse and get complementary analytical and biological information from MIBIC, NP-Atlas, and GNPS. So the MS-based ion identity network of the Rivularia crude extract revealed a big cluster with many known HP peptides such as molassamide and logopeptin A, which clustered then with putatively new HP cyclodepsipeptides that we annotated as Rivularia peptides. And one big remaining challenge for MS-based methods is now to annotate each and every compound in an extract. And recently, a collaborative effort of the Dorstein and Böker labs made a huge step forward by the development of Canopus, a deep learning-based method that assigns compound classes to every metabolite MS-MS feature in an LC-MS-MS run, including unknown metabolites. So it literally enables you to classify complete metabolites of organisms. So here's an example of a known compound from SPE fraction 1. So Canopus first correctly classifies it as depsipeptide based on the classifier ontology, and then predicts its substructures of the molecule. And with the help of CSI finger ID, the molecular fingerprints were queried against PubChem, and the query was identified as the known serine protease inhibitor molasamide. After targeted isolation and HSQC NMR acquisition in only 3 minutes, also SMART correctly dereplicated the molecule as molasamide. So now we had a lot of confidence that the annotated Rivallaria peptides are indeed new HP dipsipeptides, and there's probably also serine protease inhibitors. And isolation, structure elucidation, bioactivity assays for like 30 compounds in the fraction would probably take many months. So to create a shortcut for the most potent compounds, we established together with Daniel Petras from the Dorstein lab a method called functional metabolomics that enables us to get binding information of each and every compound in the crude extract towards the protein of interest in only 10 minutes. And after single compounds elute the column, the PA is adjusted to the optimum of your target protein. And the protein is then orthogonally infused and allowed to interact with the single molecules before acquiring a mass spectrum. So then either the protein, or in case of specific binders in your extract, the protein inhibitor complexes are measured by high-res resolution MS spectrometry. And you can see here the mass spectrum of the serine protease alpha chymotrypsine that we screened against the Rivallaria crude extract. And around 30 potential chymotrypsine binders elute then between 3.5 and 5.5 minutes and appear as additional peaks of the protein binder complex. And we first targeted the putative inhibitor with the highest relative precursor intensity with the MZ feature 1155. And next to 15 related peptides, targeted preparative HPLC yielded the new Rivallaria peptide 1155, and now we only had to elucidate its structure. So the top SMART hits were all HP cyclodepsipeptides, and many had butyryl residues, and 3 compounds in the top 10 had isolated proline residues in the side chain. But most interestingly, the top SMART hits were around 200 Daltons lighter than expected from the mass spec work, implicating a duplication of side chain modifications. And yeah, complementary, we got the molecular formula of new Rivallaria peptide 1155 from the high-res MS data, making leverage of the Sirius platform. And additionally, Canopus also revealed insightful substructural information, so it clearly hinted towards the amino acid AHP, phenylalanine, and tyrosine. But intriguingly, it also helped us with the structure elucidation of the novel part of the molecule, which are the 2 N-butyrylated proline moieties of the side chain. So finally, we were very happy to see that the new Rivallaria peptide 1155 is a picomolar elastase inhibitor and further has a 200-fold selectivity index towards chymotrypsine and more than around 100 times for proteinase K, while being inactive against trypsin. And a related compound, Rivallaria 1185, has a slightly different selectivity profile and is a double-digit nanomolar chymotrypsin, elastase, and proteinase K inhibitor, and is inactive against trypsin. Thus, the ABL unit, where is it? Here, you can see here, is responsible for the selectivity towards elastase and unimportant for proteinase K and activity. So in summary, we envisioned a new streamlined workflow for accelerated NP discovery and applied it for the investigation of a Rivallaria extract by the early integration of NMR and MS-based machine learning techniques such as SMART and Canopus to predict compound classes and their respective bioactivities, in this case, serine protease inhibition from crude fractions. So next, we screen several serine proteases versus the crude extract for new specific binders via functional metabolomics, and subsequently targeted the isolation and structural elucidation of the most promising binder. Lastly, we confirmed the bioactivity of the new Rivallaria peptide 1155 as the most potent elastase inhibitor so far known by orthogonal bioassays. Here, I want to thank all collaborators, especially the Collabora collection team and the Caro Diaz and Neyman labs, as well as the SMART and Canopus teams, and especially Daniel Petras for establishing the functional metabolomics approach. Of course, I want to thank the organizers and all of you for your kind attention. Thank you very much, and I'm very happy to discuss any questions you might have, such as what is the multiverse? Thank you.
Thank you. Thank you very much, Rafael. So there were a few questions asked in the chat, and there were responses provided. And so one question I would like to ask you is, what is the multiverse?
Okay, the multiverse, you can see here Avogadro and the mole. Now that was just a fun thing developed from, yeah, anyways, sorry. So when you look into like these organisms like cyanobacteria, they really open up a universe of molecules, and that's what we call the multiverse. And so what you can see here is the HSQC NMR spectra of 25,000 natural products. And so this space you can also like observe or search your queries. You can search your queries in the multiverse, and so also like the structures will show up, and you can like see the chemical related environment of your compounds.
Got it. And how that space is different from the space available on the Natural Product Atlas?
It is a good question. I have to compare about what Natural Product Atlas databases are in. I think the amount are similar, but like the training data set of the SMART database is mainly based on MarineLit. But we now inserted also calculated HSQC spectra to like fill in the chemical space to more than 100,000 natural products now. And they are also from plant derived, like from compounds from Dictionary of Nature, which are also in Dictionary of Natural Products. So they all cover now marine and terrestrial natural products.
So this is using the NMR spectra.
Oh, sorry. Yes.
Okay.
It is here you can see the table. So that's the only input you need is like really the H1 and 13C correlation, and then SMART automatically generates the table, and yeah, you can visualize it in this nice 180 dimensional cluster space.
Okay, yeah, so it's not using the structural similarity, but rather the NMR input as a multidimensionality reduction visualization method.
So it's a cosine similarity score between the spectra. So this is what the neural net is trained on, is the spectra here. And the more similar the spectra are, the closer they cluster together.
So there's a question by Ricardo Borges on YouTube Live. How confident are you of the calculated HSQC spectra?
Yeah, that's a great question. We still validate that more, but we are very confident. Unfortunately, I forgot to put in the slide, but there will be a new version called SMART 3, which is based on molecular fingerprints. And there we have all the validation available, and the calculated data almost equally performs as the real data. And if you combine them, you get even better results. So that makes us very confident. But that might differ from case to case and also from what solvent you use. But in general, it seems to be quite robust to different NMR solvents.
Yeah, and they did a follow-up question. How detrimental is it if there are some low confidence predicted chemical shifts that are used in the comparison?
So I would always recommend to do as much as possible, because the analysis time is like 8 seconds. So if you are on Mastronova or Topspin, you can just, for example, first like label everything there is and test what it is like, and then maybe have a more knowledge look and go into details, and you hopefully will be astonished how similar the results is. So sometimes I put in solvents like DMSO peaks, I'm too lazy to remove them, or any noise peaks. So additional and lacking correlation, if not crucial for the molecule, should not hamper the results by much.
Okay, and there was one question from Mehdi about the new compound that you isolated. Was the absolute configuration established? If yes, by which method? And how many milligrams or micrograms of the compound was isolated?
Yeah, both great questions. So for the first question, we did not determine the absolute configuration yet, but it is very well established. So there's like 200 HP cyclodepsipeptides known, and the amino acids are almost, I think exclusively configurated. But you can easily do that with advanced Murphy's analysis. So that should or could be done. And the second question was the quantities. So the compound here was the highest quantities, which is a little bit unfortunate. It was 1.6 milligrams. It's an environmental sample that we cannot cultivate. So we have a lot of compounds ranging in like 50 micrograms to 0.5 milligrams. So the structural elucidation of those is a little bit more challenging, but possible.
Okay, thank you very much.
Thank you very much, Rafael. Thank you, the speakers, for the great section. We are a little bit over time. I'm sorry if you had like an unanswered question. We will have the breakout rooms after the last section, and then you'll be able to ask the questions directly for the speakers. Thank you, everybody, for the section. And now we are going to the next session, the Flash Talk session.
Thank you very much, Ricardo. So my name is Julia Gauklitz. I'm currently in the Dorstein lab, and I'll be co-hosting this session with Laura Isabel McCall. Laura Isabel, would you like to introduce yourself briefly?
Yeah, thanks, Julia. I'm Laura Isabel McCall. I'm assistant professor in the Department of Chemistry and Biochemistry at the University of Oklahoma. And my lab uses molecular networking to understand the relationship between location and disease.
Awesome. So I just want to start our Flash Talk session by thanking you all for participating in this first GNPS user meeting. And we will be continuing with a geographically and scientifically diverse set of Flash Talks, where you'll get some brief insights into tool development, environmental samples, as well as investigations into disease. So we're really highlighting a broad set of science that can really benefit from the GNPS infrastructure. I will share my screen where I have all the Flash Talks located. And our first presenter is going to be full screen mode, Christian Martin. And we just need to unmute him. Laura Isabel, could you do that?
Yeah.
Oh, perfect.
Can you hear me?
Yes, please go ahead. Welcome.
Okay, thank you, Julia. Thank you, Laura. Good evening, guys from Panama. Today I'm going to talk about viscosine-like lipopeptides from frog skin bacteria that inhibit Aspergillus fumigatus and Patecocytium dendrobatidis. First of all, I'm sorry.
Sorry.
Okay. As an amphibian, have been worldwide affected by a pathogenic fungus called Patecocytium dendrobatidis. This study was focused on understanding how the cultivable fraction of epibiotic bacteria in Panamanian frogs would help us to find out antifungals against Aspergillus fumigatus and Patecocytium dendrobatidis, or BD. To do that, we sequenced more than 200 isolates and tested them against Aspergillus fumigatus. And in this experiment, we realized that Pseudomonas cori produced the highest inhibition zone against this fungus, Aspergillus fumigatus. Then we replicate the same experiment, but now using multi-imaging mass spectrometry and found that the molecules in the bacteria and the inhibition zone had molecular weights over 1,000 Daltons. Then we did molecular networking and identified a cluster of cyclic lipopeptides composed basically for viscosine and macetolides, whose masses matched with the multi-imaging experiments. So then we tried to, actually we managed to separate viscosine by HPLC and compare it and did NMR experiments too, and did also antifungal tests against Aspergillus fumigatus and DG. And in this part of this work, we reported by first time a scientific inhibitory effect of a bacterial cyclic lipopeptide from frog skin against Aspergillus fumigatus, which is a human opportunistic fungus, and Batacocytium dendrobatidis, which is the main pathogen of amphibian in the world. So if we take a look of this fungus BD represent to amphibians what coronavirus represent for humans now. So just to give you a brief idea what important is our findings. Thank you.
Thanks, Christian. So our next poster is by Robbie Quinn. Go for it, Robbie.
Hi, I don't know if you can you hear me?
Julia, can you switch the slide?
It's switched. Can you hear me?
Yes.
Can you see me? Because I'm wearing a Hawaiian shirt for a more authentic experience.
We can see and hear you. It's awesome.
This is the most dressed up I've been since March, so it's a special day for me. What I'm going to talk about today is an interesting finding that we did with some collaborators at the Hawaii Institute of Marine Biology. They were studying coral bleaching, essentially, in Kaneohe Bay in Hawaii. In 2015, there was a severe bleaching event. So briefly, coral bleaching is basically kind of a non-permanent disease of corals where their symbiotic algae leaves their cells, and it can be driven by high water temperatures. So in 2015, in Kaneohe Bay on the east coast of Oahu, there were basically record high water temperatures, and what they say is the worst bleaching event anyone's ever seen in Hawaii. Interestingly, the bleaching was not universal. So you can see in the picture here, two corals physically adjacent to each other. One has a bleached phenotype and the other does not. And we don't totally know why. A lot of it has to do with the algae that they host in their cells. So what was done back then, before I was involved with this group, they basically tagged the corals that bleached and those that didn't. And then 4 years later, which is kind of in spring and winter of last year, I got involved in some of this work, and Ty Roach at the Hawaii Institute of Marine Biology sampled these corals, essentially took nubbins of the corals, and we were basically studying the bleaching history and the metabolomics differences that may exist in these corals. And an important point is that when they were sampled, they were physically healthy. So they're not bleached at this point. They're basically indistinguishable visibly, but they have this historical bleaching phenotype. So sure enough, the LC-MS-MS metabolomics, GNPS analysis, feature-based molecular networking, all of those fancy terms, ultimately give us this first look at the data that I'm showing in the kind of orange and red, blue plot there, is there was a strong signature of the phenotype of the bleaching history in the corals 4 years after they bleached. So there were no significant bleaching events since then, but even 4 years later, you could easily distinguish the corals that had that historical bleaching phenotype even when they're actually healthy and not bleached. So that was pretty interesting to us. We used some machine learning methods to kind of pull out which molecules are mostly responsible for that difference. Many of them were unknown. We looked into some older literature about algal lipids, which we know a lot about because people study algae for biofuels. And it turned out that the major signatures, major metabolites that were driving that historical bleaching phenotype were what are called betaine lipids. And I show the molecular network there at the bottom. The nodes are actually colored. It's a bit hard to see, didn't come out too well, but by whether they were from the bleached or non-bleached corals. What you can see is these betaine lipids, which I'm showing you on the right, they're phosphorus-free lipids. So they're only found in algae. They're found in Symbiodiniaceae type algae, which are the symbionts in the corals. And what we found, you can really see it in the molecular network, which really shows you the beauty of this approach, is the biological difference between the bleached and non-bleached corals kind of cascades through that network. So in the top left, you see a bunch of nodes that are very gray, which means they're found mostly in the non-bleached corals. So we say the resistant corals. Those betaine lipids, the structure of them was basically a single acyl chain that was completely saturated or mostly saturated. Some of the features were maybe 1 or 2 saturations. Further away in that network, if you move kind of southeast, you see a lot more white, which is those were molecules that were primarily found in the bleached corals. Sure enough, those are highly saturated, often diacyl lipids. So the biggest difference in the history of a coral's resistance to bleaching are these betaine lipids in the algal symbiont. And literally, the chemistry specifics is whether or not they're fully or more unsaturated, can tell the difference between whether or not these corals survive. And we think there's a lot of kind of coral reef restitution opportunities here that we could figure out what algae have these lipids and then possibly plant them on the reef to help the reef stability. The final point I'll make is that we also did a temperature stress experiment. There were many other molecules that reflected this bleaching history, some of which were from the coral animal themselves. What you see is that this phenotype is so deeply imprinted in the corals that even under temperature stress, they don't become more similar to each other. They actually become even more different. So that different phenotype of the corals is there when they're normal and not bleached. We stress them and that phenotype remains. They move in a similar direction, but they don't become more similar to each other. They always seem to stay different. So this bleaching history is deeply imprinted in the coral metabolome. I have no idea how long I just talked for.
All right, Robbie, thank you so much.
You're muted,
Julia, you're muted.
Okay, thank you very much. Next up, we have Mabel Gonzalez.
Hi, can you hear me now?
Yes.
Okay. Hi, everybody. So my name is Mabel Gonzalez, and I want to show you a little bit from my work, which is the application of GNPS for standardizing a non-lethal method for analyzing the secretions from poison frogs from Colombia. So amphibians are really great because they have a huge chemiodiversity, only comparable maybe with plants and microorganisms. But the drovatids, the unique chemistry that they have is only found in the frogs and also the invertebrates that they eat. So the specific superfamily that I am working on is called Dendrobatidae. And the problem is this, that the usual gold standard method for analyzing the secretions from the frogs is lethal. So involve killing the frog and removing the skin. So that's the specific target that I was interested in solve. So what I was using was 2 different species. One is critical endangered species from Colombia, and the other one is in a least concern status. And what I decided to do is to use swabs, just for swabbing the skin from the frog, and comparing the extraction efficiency of 3 different materials, rayon, cotton, and polyurethane foam, and comparing it with the gold standard method, which involved the skinning of the frog and posterior methanolic extraction. So the workflow that I used, you can see it in the screen, and it was using metabolomics, GCMS metabolomics. And at the end, with the GNPS output, what I was using, what I was able to see, it was seeing the different capabilities of the different extraction materials with the specific chemistry of the frogs. So in this specific cluster, for example, which is an indolecithin cluster, which is one of the types of alkaloids that the frogs secrete, you can see that some specific compounds or nodes are equally extracted for the different 3 swab materials and also for the gold standard method, but other ones seems to be extracted differentially. So I was able to see here using GNPS is that probably the best solution for the frogs is like using a different combination of materials for swabbing the skin and not only one of them because of the porality differences. So also with the output from GNPS, I was able to recognize that the 3 materials was able to sample the different chemiodiversity from the two species of the frogs, and actually it was able to extract the different chemiodiversity found in the same species but in two different locations, which is really important because many efforts for biodiversity purpose and conservation are targeting only the phylogenetical information, but not like the location. And this chemiodiversity differences shows how important it is to protect the species in different locations because the species are different. Also, the genetic information, of course, is different, but also the chemicals are different. So this is really, really helpful for doing that. And this is a great measure for the frogs because we have now a new method that we can use directly on the fleet, directly on the field, and release the frogs without harming them, and just sample them and just releasing directly on the field. So that's really nice. Thank you.
Thanks, Mabel. Next, we have Jessica Montoya.
Hi, can you hear me? Hello.
We can hear you, but not very well.
Okay.
Can you get closer to your computer? Sometimes if you put your face closer to the computer, it will give better sound.
And now?
That's better.
Okay. Okay. Hello, my name is Jessica Montoya, and I want to show you the results of a project developed by CBOP, a research group from EAFIT University in Medellin, Colombia, under the direction of Valesca Villegas Lab, and in collaboration with the Dr. Mauricio Caraballo and PI Peter Dorsten from UCSD. So the bacterial strains Bacillus tequilensis EACV 0015 is an active ingredient that reduces the severity of different plant pathogens. However, when this strain grows in liquid or solid culture, a new morphotype starts to emerge frequently. This new morphotype was isolated and identified as Bacillus tequilensis by DNA sequencing. This new morphotype, called M1, shows phenotypic difference related to wild-type strain, such as a reduce of antifungal activity and biofilm formation. In order to get insights to the reduce of antifungal activity, an untargeted LC-MS-MS-based metabolomics approaches was performed to compare the metabolic profile of both morphotypes in different growth phases. For this, we use MC-MIND2, GNPS, and Cytoscape. As a result, in the PICO-A plot, we can observe the grouping of different clusters of samples, one of which indicated that the set of features exclusively belong to wild-type strain in mid-log phase and in transition phase. Other results that we found that of the 2,142 features detected in total, 214 features were detected only in wild-type strain and 36 features in M1 morphotype. Finally, we identified that the families of angisins and iterins lipopeptides were only detected in wild-type strain, and surfactins lipopeptides were more abundant in wild-type strain than in M1 morphotype. So with this result, we conclude that the metabolic profile of both morphotypes are different. Thank you.
Great, thank you very much. Next up, we have Tiago Leal.
Hello, everyone. I'm Tiago Leal, a postdoc from the Dorstein Lab at UCSD, and thank you for the opportunity to speak here today about the multiomics profiling of tropical marine cyanobacteria. So in the left, we use the presence-absence of biosynthetic genes to predict beta diversity, and then to highlight the most interesting strains for genome mining. In the center, we apply GNPS monad enhancer to confirm that the metabolic classes were consistent with the classes that we observed in the genomes. We also observed several genus-specific metabolites in the monad enhancer result. Last, in the right side, we wanted to connect genomics with metabolomics via KNN machine learning fingerprinting approach. Despite the small sample size, we correctly predicted the biosynthetic genes for 1 out of 8 compounds, Curacin A being that compound. And now we are going to apply the same machine learning approach to a bigger data set from the Pair Omics data platform by Dr. Marnix Medma and collaborators. Thank you.
Thanks, Tiago. Up next, we have Gordon Liu.
Hi, there we go. Hey everyone, my name is Gordon, and thanks for the chance to speak here. I'm from the Sanchez Lab at University of Illinois at Chicago. And today I'm going to be talking about Blanca, which is a tool that we developed in the Sanchez Lab to help with removing blank signals from our data sets. So when using very rich media, very rich chemically undefined media, such as the cheese curd agar that we use, we found that a lot of times our extracts contain signals that correspond to media components that convolute our downstream analysis, such as analysis using GNPS. So as an example, on the left is one of the larger clusters from a network that was made using GNPS. For these nodes, in red is our cheese curd agar, blue is a fungal extract, and green is that same fungal extract that was processed with Blanca. We see that in these nodes, especially the ones that are red and blue, there are a lot of media components such as linoleic acid, which is a fatty acid that's related to dairy products, or 9-octadicenamide, which is a food preservative. And so using Blanca, we can remove these spectra from the raw MZXML files that we get from our mass spec analysis, and removing these based on spectral similarity, we can then have a simplified data set. And in the case of the simplified data set, which is seen on the right, you have much fewer nodes, and although some media components may slip through, it's a lot easier to analyze these simplified data sets, which only have a handful of signals, versus the dozens or maybe even hundreds of signals that you would have in larger experiments. And so just on the bottom there, I want to point out that the link to all my code is in the bottom right there. Thank you.
Thank you. Next up, we have Danya Dean.
Yeah, so I'll be presenting some of my work entitled Spatial Identifies Chemical Changes in Chronic T. cruzi Cardiac Tissue. So Chagas disease is a neglected tropical disease caused by the Trypanosoma cruzi, and it occurs in 2 stages, an acute and a chronic stage. My study focuses on the chronic stage of the disease. During the chronic cardiac Chagas disease, it's associated with severe symptoms such as arrhythmias, cardiac failure, and apical aneurysms. And unfortunately, Chagas disease pathogenesis is not fully understood. So we present a spatial metabolic approach to help us understand CD pathogenesis, where we hypothesize that chronic T. cruzi infection causes localized metabolic alterations in cardiac tissue. So what we did was we infected mice with either CL or Silvio X10-4 strained T. cruzi trypanomastogotes, and after the chronic stage of infection, we euthanized the mice and collected the heart tissue, and we sectioned that and then ran that on LC-MS. And then from that, we were able to use GNPS to help us annotate specific chemical families associated with infection. Specifically in figure 4, we were able to see that chemical families, as seen in figure 4A, which represents a subnet of azocarnitines, and B represents a subnetwork of phosphocholine, where red represents CL, gold represents Silvio X10-4, we see that these specific chemical families were altered by infection and were associated with those specific strain types. Additionally, in figure 2, we see that total azocarnitines were characteristically decreased by infection and seem to be specifically significantly different in certain positions of the heart, as seen in Silvio X10-4 in positions B and C, and total phosphates were characteristically increased by infection and found to be statistically different in C of CL. So overall, our study was able to help us identify positional metabolic differences associated with T. cruzi infection and show the strength of systematic chemical cartography to help us understand disease tropism. Thank you.
Thanks, Dania. Up next, we have Advaita Parab.
Thanks for the introduction, Dr. McCall, and the organizers for giving me an opportunity to speak today. I'll be discussing results from an untargeted metabolomic study of mice that were infected with the parasite Leishmania major, which is the causative agent for a neglected tropical disease called cutaneous leishmaniasis. It affects about 12 million people worldwide. And so for the experimental setup, we had 5 mice infected in the year and 5 mice infected in the foot pad with tissue samples obtained at 7 and 8 weeks post-infection, following which LC-MS-MS was performed on the samples, and untargeted metabolomics allowed us to identify specific metabolites that were changed with infection in this model. And so using GNPS, we were able to make annotations with confidence using parameters such as the cosine score, ppm error, and also looking at the quality of mirror plots. And so among the top metabolites identified were omega-3 fatty acid eicosapentaenoic acid, tissue phospholipids, specifically phosphocholine, as well as amino acid glutamine. Furthermore, molecular networking allowed us to annotate metabolites that belong to the same chemical families with the possibility that they served a similar biological function. Interestingly, we were able to identify specific metabolites that were increased with infection in lesion adjacent sites, which appeared macroscopically healthy. Additionally, we observed opposite trends for different masses of phosphocholine, with the lower and higher mass ranges showing opposite trends with infection. All in all, untargeted metabolomics provided some insights into the molecular level changes occurring with infection and biochemical pathways that are important to the disease pathogenesis of leishmaniasis. And so future studies could definitely build up on this data, and studies looking at disease interventions could start looking at specific pathways that are important to CL pathogenesis. Thank you.
Great, thank you so much. Last up we have, Allegra Aaron, but there's an issue here. Nope. Okay.
Maybe Allegra can share it herself. I think she
Yeah, if it's easier, I can probably share my screen.
Open as a separate one as well.
Oh, okay.
Okay, I don't know what's going on. Go ahead and share. I'll stop share.
Okay, give me 1 second. Oh no, my share screen is disabled.
Daniel, can you add her as a co-host? Okay, you should be able to now, Allegra.
Okay, great. Okay, can everyone see that?
Looks great.
Okay. Okay. Okay, so thank you for the opportunity to talk. I guess I'm the last person talking, so thank you as well for sticking around to listen. My name's Allegra, and I'm a postdoc in the Dorstein lab as well. So while untargeted metabolomics is a powerful tool for the discovery of new molecules, it does not provide functional information about these molecules. And we actually just heard about a new strategy that was called functional metabolomics in Rafa's earlier talk. But in addition to ligand binding, metal binding is also a critical molecular function. Now, current mass spec-based strategies for elucidating metal binding properties rely on metal isotope patterns, which can really only be applied to certain metals, and they're also challenging to apply to complex samples. And so with this challenge, we developed a two-step native electrospray ionization mass spec method in which post-column pH adjustment with either a base or a buffer and a metal infusion with either one or a mixture of metals are combined with ion identity molecular networking, which is a rule-based informatics workflow recently developed in our lab. So this method identifies metal binding compounds based on defined M/Z offsets of ion features with the same chromatographic profiles. And so we showed that this method is both specific and selective. So only known siderophores are identified as iron binders in standard mixtures. And then different siderophores actually exhibit different metal binding preferences when a mixture of metals, including iron, copper, cobalt, and others, are infused. Now, based on this preliminary data, the method was applied to both bacterial and fungal culture extracts. And we found both known and novel siderophores in the two extracts. And finally, we were able to apply this method to dissolved organic matter samples, which was really exciting because these samples were extremely complex coming from the California current ecosystem. And our method actually revealed that domoic acid binds copper as a dimer. Now, based on its easy and flexible implementation, we believe this method has the potential to become a really useful strategy for elucidating metal binding molecules in complex samples of all types. And we encourage all of you to try it out actually on your own instruments. So yeah, I think that's all for me.
Great, thank you, Allegra. Thank you to all of the Flash Talk presenters for contributing your science to this meeting. And there is not time for discussion now as a large group, but we will be breaking out into different individual Zoom calls where you can then go in and out to ask questions for the different sessions. And I'll hand it over to Daniel for final words.
Hey, thanks a lot, Julia, and yeah, thanks a lot to all the speakers for this awesome talks, and all of you for stopping by and making this possible. Yeah, I think we're close to the end, and I just posted the links for the 3 breakout rooms in the chat. They are also in the email you should have gotten yesterday. So yeah, I hope that those breakout rooms will enable basically a more like informal conversation with the speakers or with like different participants. And yeah, I hope to see you there.
Oh,
if not,
can I ask one question?
Next time.
Can I ask
Sure,
a question about the breakout rooms? So if we talked in one section, but we want to ask questions in another section, can we like go back and forth from breakout room to breakout room?
You can go back and forth. That's why we have them as 3 different Zoom calls actually, and not as like built-in breakout rooms.
Okay,
so yeah,
cool.
just feel free to hop between them. I would encourage the speakers to stay in the breakout rooms of their sessions so people can find them, but maybe towards the end, they can also start moving like you would do in real physical.
In a real poster session. Thank you.
Unless Ming wants to say some closing words, I think that's pretty much it. And thanks again for coming, and hope to see you in the future or in the breakout rooms now.
Yeah, definitely. Thank you all for coming. And again, the GNPS community would not be anything without you all. And as we've seen kind of throughout the last couple of years, it's always growing. And so it grows and gets better through feedback, new tools. And so I think it's crazy. It's before I think we were excited to kind of build GNPS and kind of build out what we thought was what the community needed or wanted. But nowadays, the computational community especially is moving so fast, we're just trying to keep up. So that's a great thing to have. That shows the field is moving, and you all are a big reason for that. So anyway, thank you again. And thank the organizers that actually did all the hard work and the presenters. It was a pleasure.